Hi, this is Haley Marks, and for some reason the Zoom website isn't allowing me to record sound, or there's something wrong with my computer audio, so I'm gonna record this on my phone and see if that works better. So this is my presentation for the Photosynthesis Hill Reaction Lab. For this lab, it's important to understand the concepts of photosynthesis, which is shown in the leftmost picture, light reaction, which is shown in the right bottom picture, and then the Hill reaction, which is shown in the top right picture. And overall, the Hill reaction and light reaction are a part of photosynthesis, which is the bigger process in this lab. So photosynthesis, simply put, occurs within chloroplasts and some algae and bacteria. And it happens when water and light energy combine, and along with carbon dioxide, it creates glucose or other similar large organic molecules. And it occurs within two parts: the light reaction, which is again shown in the picture on the bottom right, and dark reactions, or the Calvin cycle, which I won't be talking about in this PowerPoint presentation. So the light reaction happens when chlorophyll absorbs light, which is shown in photosystems 2 and photosystems 1 by the yellow lightning bolt thing, and it happens in the thylakoid membrane. So the high energy electrons are released, and those electrons move through electron carriers, which again is in the thylakoid membrane. And in photosystems 1, NADPH is produced, and then through in the ATP synthesis red part of the diagram, ATP is produced along with H+ ions. The production of NADPH shows that the light reaction is essentially completed, and the energy also results in ATP production. The Hill reaction, shown in the top, is again part of light reaction, and during this stage, the acceptor goes into the chloroplasts, and when it is released, it is reduced because the electrons from the water that also go into the chloroplasts are transported to the acceptor, and oxygen is released, which is also shown in the leaf part of the photosynthesis, which is again the main point of photosynthesis, to produce oxygen and glucose. In this lab, the Hill reaction was mainly measured using isolated chloroplasts from spinach leaves, and one of the products used was DCIP, which is an acceptor, and when it is reduced, it changes from blue to colorless. DCIP was used in all three tubes to show when it was reduced. DCMU was only used in one tube and is an inhibitor of photosynthesis. What happens is it blocks plastoquinin from binding in photosystems 2. The spectrophotometer was used to measure the absorbance of each tube throughout a time of 10 minutes, and it essentially measures the intensity of light or photons as it passes through a solution. The tris NaCl buffer was also used in all tubes, and it maintained the pH of all the tubes within a certain range. So if we had been at Cortland and done this experiment, first we would have isolated chloroplasts from spinach leaves, and then we would have created a blank, and all three tubes along with the blank can be created from the table shown at the top of the page. So for tube 1, once it was made, we had to initially measure it, and then 10 minutes later measure again. So it was only measured 2 times throughout the experiment. For tube 2, an initial reading would be taken, and the absorbance would be measured every minute for 10 minutes. That would be the same for tube 3, except for that DCMU would be in tube 3. And when not being measured for its absorbance, the tubes would need to be kept in foil or in front of the light. And once all of the data was taken, an analysis would be made, which is shown in this graph from the sample data we were provided. The blue line and dots shows the foil wrap tube. The standard in the experiment is shown in the orange reddish dot. And then the gray shows tube 3, the DCMU. And the equations of each line and the R-squared values are also shown. So for the standard, the absorbance decreased within the 10 minutes of the experiment. For the blue, it decreased slightly, but overall mostly remained the same. And for the DCMU, it remained constant, which can be shown by its equation, because there's no x value in it, and it's just a single number. For the results analysis, we can start with the standard. So the standard decreased in absorbance because it was placed in front of the light throughout the entire experiment, and there's no DCMU, nothing that would inhibit photosynthesis. So the chloroplasts in the tube were able to use that artificial light and carry out photosynthesis to create glucose, energy, and oxygen. So that glucose, energy, and oxygen built up within the tube, and so when it was placed in the spectrophotometer, the light within the machine wasn't able to pass through the tube as easily as it was in the beginning of the experiment, so the absorbance decreased throughout the experiment. For the foil, it mostly remained the same because it wasn't exposed to the light energy, and it didn't have any resources to help it go through photosynthesis. And the current energy that was in the tube before the experiment started didn't have anywhere to go, so it stayed in the tube, which is why the absorbance mainly stayed the same, but slightly decreased. The DCMU remained constant throughout the experiment because, again, it inhibited plastoquinin from binding in photosystems 2. And essentially, what DCMU does is it doesn't allow electrons to flow from photosystems 2 to plastoquinin. So it wasn't able to produce any energy or glucose or oxygen that would normally be produced in photosynthesis. So the main difference between DCMU and the foil, even though they both mostly remain the same, is that the DCMU, it was exposed to the light and energy, same as the standard, but the DCMU inhibited the electrons, which can be shown in the diagram on the screen. So it had the energy, but it wasn't capable of using that energy to produce anything. The foil had that energy, the foil didn't have that energy from the light, and over time, the current energy and oxygen slowly faded away or faded out of the tube, which is why the absorbance slightly decreased. When exposed to light in between when reading, the chloroplasts in the foil were slightly able to produce energy and oxygen and glucose with the light from above. So it was able to produce a slight amount of photosynthesis, which is why it slightly decreased, as shown by the equation for the blue at the top right, while the DCMU completely cut off photosynthesis and didn't allow the reaction to carry out at all, which is why there's no increase, there's no decrease. So, and the DCIP used in the tubes, when it was oxidized, it was blue, and then when it was reduced, it was colorless for all of the tubes. So, thank you.